Mendel's experiments opened the door to understanding inheritance, but many questions remain. In the early part of the 20th century, we didn't know the nature of these factors that Mendel talked about. These next steps involved many researchers in the early part of the century, and they aided to unite information about the behavior of chromosomes. The behavior of chromosomes during meiosis not only explains Mendel's principles, but leads to a new and different approach to the study of heredity. The central role for chromosomes in heredity was first suggested in 1900 by a German geneticist called Karl Correns. He suggested this in one of the papers announcing his rediscovery of Mendel's work. Soon after, observations that similar chromosomes paired with one another during meiosis led to the chromosomal theory of inheritance, which was proposed by an American scientist named Walter Sutton in 1902. Shortly after that, in 1910, Thomas Hunt Morgan was studying the fruit fly, Drosophila melanogaster. He discovered there was a mutant male fly with white eyes instead of red eyes. And he thought this was curious. So he crossed the mutant male to a normal red-eyed female and found that all the F1 progeny were red-eyed, which illustrated to him that it was a dominant trait. Morgan crossed some F1 females with some F1 males and found that the F2 generation contained red and white eyes. But white-eyed flies were male. So this suggested to him something about that some genes must be sex-linked. When he test-crossed the F1 female with a white-eyed male, it showed the viability of white-eyed females. That is, the possibility that we could actually get white-eyed females. Morgan concluded from these experiments that the eye color gene resides on the X chromosome. If you examine these figures over to the right, you can follow that thought pattern. If we have the parental generation female and then a white-eyed male, and we cross these two individuals, you'll see the same heterozygous condition with red eyes as dominant. However, when we cross the F1 generation female with an F1 generation male, we see that 50% of the males have white eyes. Looking at the test cross, we found that the female carrying one copy of the white eye paired with that male could also produce white-eyed females. And traits determined by genes on the X chromosome are said to be sex-linked or X-linked because it's associated with the sex of the individual. Morgan's experiment was one of the most important in the history of genetics because it presented the first clear evidence that the genes determining Mendelian traits do indeed reside on chromosomes, just as Sutton had proposed. Mendelian traits segregate in genetic crosses because homologous chromosomes separate during gamete formation. The structure and number of sex chromosomes vary in different species. In the fruit fly, Drosophila melanogaster, it's based on the number of X chromosomes. So if an individual has 2 X chromosomes, no matter what other chromosomes it has, it's a female. And the male will have 1 X and 1 Y. In humans, we have a similar situation. However, an individual is female if it has 2 X chromosomes and only 2 X chromosomes. There are a few anomalies, but in general. A male has a Y chromosome, so it's X and Y for the genotype. Humans have a total of 46 chromosomes. As you know, 22 pairs are autosomes, and 1 pair are the sex chromosomes, either XX or XY. The Y chromosome, you'll notice by looking in the picture above, is tiny. It's because it's highly condensed, and there's really not that much on it. Or you could say that the X chromosome has all the baggage, I don't know. So this is the reason that sex-linked traits show up more frequently in males, because recessive alleles that are on the male's X have no active counterpart on the Y for possible covering of the recessive trait. Now, sex is determined by a region on the Y chromosome called the SRY gene, sex-determining region of the Y chromosome. If the SRY gene is present, which it usually is on the Y chromosome, then SRY gene is expressed, and that determines that testes develop, and thus testosterone, and all the things that are associated with maleness. However, if the Y chromosome does not have the SRY gene, then there's a possibility that you could have an XY female, because there's no SRY, and thus the default is ovaries, and there's no testosterone, and thus we end up with a female that has the genotype XY. There's some really interesting sexual deviations that we see throughout development. A fascinating field, I think. Perhaps we'll address some later. But getting back to the point, some human genetic disorders display sex linkage. Of course, we've been noting conditions throughout time that seem to affect males to a higher degree than females. For example, red-green color blindness is one we know well. Many more males are affected by it than females. This is because the gene affected is carried on the X chromosome, and all it takes is one copy of the recessive allele and no coverage by another X chromosome, and we have the condition. Hemophilia is another great example. It's a disease that affects a single protein in a cascade of proteins that are involved in the formation of blood clots. And the allele for hemophilia was introduced into a number of different European royal families by Queen Victoria of England. One of the forms of hemophilia is caused by this X-linked recessive allele. Let's look at all the possible genotypes. Let's say we have a female who has one dominant copy and one recessive copy. Let's say we have a female who has one dominant copy and one recessive copy of the allele. She is a female carrier. Let's say that a female has both dominant copies. She would be termed unaffected. If she were to have both recessive copies, then she would be termed affected. So these are the three possibilities for a female. If she has one copy of the recessive allele and still a functional copy in the dominant allele, then her blood is able to clot just fine and she does not suffer from hemophilia. It takes two of the recessive alleles in order for her to be affected. Now let's look at the male genotypes. Males have one X and one Y. If the X has a recessive allele, the male is affected. The other option he has is he could have the dominant allele and a Y, in which case this male is unaffected. So while we're thinking about this notation, take a moment to cross an affected male with a female carrier and see what resulting phenotypes you end up with. Here's the pedigree that illustrates the relationship between European royal families. This is where the condition first shows up for Queen Victoria. One of her daughters was a carrier and married Tsar Nicholas II. This is how it came into the Russian royal family. Although males only have one copy of the X chromosome and females have two, females don't produce twice as much of the proteins that are encoded by the genes on the X chromosome. Instead, one of the X chromosomes in females is inactivated early in embryonic development. This inactivation is an example of dosage compensation. It ensures an equal expressions of genes from the sex chromosomes, even though females have two X chromosomes and males have only one. In each female cell, one of the X's is inactivated. You can see this illustrated here on the right. One of the X's becomes what's called a bar body, a very highly condensed X chromosome. Females heterozygous for genes on the X chromosome are genetic mosaics. Calico cats are a perfect example. Have you ever seen a male calico cat? It would be very, very rare and for different reasons. Here's what happens. In certain patches, the red allele is turned on and the black allele is inactivated. In other cells, the black allele is on and the orange allele is inactivated. It's inactivated because it's coiled up so tightly as a bar body. All the subsequent cells that result from division of this cell will have orange color to them. And all of those that develop from this cell will have black. This explains the patchiness we see in calico cats. There are some exceptions to chromosome theory. Not all of the genetic information that we obtain from our parents comes from the nucleus. As you'll recall from our chapter on cell structures, both mitochondria and chloroplasts have their own DNA and thus their own genes. Traits that are controlled by these genes do not follow the chromosomal theory of inheritance. For example, genes from the mitochondria only come to us in the egg, and thus our father does not contribute any mitochondrial DNA. The same thing occurs in plants with their chloroplasts as well as their mitochondria. This is called maternal inheritance, and it can be used to trace some evolutionary history. As in animals, plants also have chloroplasts that are often inherited from the mother. However, it does vary from species to species. So some species will get mitochondria and chloroplasts from both parents, or only from the father, or only from the mother, but usually from the mother. Take a moment here to look at some of the problems with sex-linked inheritance and practice doing monohybrid crosses and predicting the outcomes of crosses with X-linked characteristics.